You can give a disembodied lab-grown brain from human stem cells synesthesia. Researchers are training them to experience the world, and it's leading to a lot of existential questions about how we experience it. Now, a lot has happened since researchers first grew the tiny disembodied brains in around 2012, although there's evidence they may have been grown as early as 2007. That's just from my own investigations of DARPA funding. Essentially, the only sensation that they have that's ingrained is going to be sight, and that's just going to be the reception of light. When light shines on their little eyes, then they do have some brain activity responding to it. The reason they have eyes is because your eyes are actually part of your nervous system, and they develop from the same stem cells when you are developing. Now, if you really wanted to give them additional ingrained sensation, you could combine different organoids. You can even give them skin. That image is demonstrating combining them with vascular organoids. You can even give them a tiny organoid heart. Although largely the issue of them becoming hypoxic is gone, researchers have figured out how to keep them alive for 5 years or more. Now, their development tends to follow at least the gene expression you would expect of, say, a 5-year-old, but they still end up tiny. You kind of need a body if you want a fully functional brain. I also don't know why the concept of giving them skin bothers me, but yes, you can do that too. But you can also give them touch, despite the fact they don't really have the nervous system required for touch. Researchers have put them in all sorts of video games and robots. The first one that was quite publicized was a spider robot. But they've also been placed into Pac-Man and a butterfly simulation. However, they do need to experience the world in order to engage with it. In this study, they gave them tactile sensation so that they could read Braille. To do that, they hook them up to electrodes, and those electrodes can measure the output that they give, but they also give them electrical shocks. We do know that they have a preference. They don't like disordered electrical shocks. However, they can associate something like a tactile sensation with a certain pattern of shock. And yes, they can learn to operate robots with a kind of vision. Different kinds of sensations give different patterns, and they perceive those and are able to move in that world. Yes, they're receiving the same kind of input in various patterns, and they can associate that with a reaction. So far, we've largely seen that for things like movement, although giving them sensation is becoming more common. This paper had an interesting proposal. For epilepsy research, they can give mice the condition and then see how they behave. Epilepsy can interfere with people's memory and the way that their neurons interact with each other. That can be seen with a mouse, sure, and it can be compared to a standard mouse, but it's still not a human. But what if we took human brain organoids and used that to run a simulation of a mouse? And then we can model the ability for that brain organoid to learn and interact with the environment as an analog to people. So dropping a brain organoid in a mouse simulation. Yes, that's kind of the very basics of what we would need to ultimately get the matrix. Convincing a human brain they are actually a mouse. Now, of course, I should provide all of the disclaimers. These are little blobs of goo, not actually a fully functional conscious human brain, but a component of it. It could be part of a human brain only if you transplant it into a human brain. Otherwise, they get transplanted into, say, stroke-induced mice, and we just call them a mouse. Now, this is an extraordinarily reductionist view. When we think of our brain sets all that we are, you can have a head injury and your personality will change significantly. So what does it mean to be a tiny subcomponent of something that could be a brain, and what kind of responsibilities do we have to maybe not put them in a mouse simulation? I don't have the answers to many of these questions, but it does make me think of the brain in the vat, the philosophical brain in a vat. How would you know if you're experiencing reality if all of your input comes from the outside? And that's what we're already living. So we experience things like touch and smell and taste because electrical impulses are processed by our brain, and that gives us a sensation. But that also breaks down for people with synesthesia. They aren't perceiving the world in the same way that others are, and I'm not entirely sure if any one way to experience the world is correct. So when I think of brain organoids learning sound and touch and taste and smell, I'm really not sure how different our experience is in that regard. We're just much more complicated, more integrated organoids. Yeah, it's kind of existential, and I'm still deciding how I feel about it. Follow for more man-made horror!